Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And in the current module, we are discussing about the genetic material. What we have discussed so far, we have discussed about the discovery of genetic material by doing the different types of experiments. So initially, there were two prominent candidate for the genetic material. One is the nucleic acid, the second is the proteins, and there was a long debate about which material is actually going to serve the purpose of having a potential to be genetic material. We have discussed about the different types of experiments through which the scientists have proved that it is actually the genetic nucleic acid which is actually been the most acceptable material for genetic material. So within the nucleic acid, it could be DNA or the RNA. Mostly the cells are actually having the genetic material in the form of DNA, whereas in some of the organisms such as viruses, you are also going to have the RNA as the genetic material. Then in the previous lecture, we have discussed about the genomic organizations and how the genetic material is actually going to be organized in the different types of organisms. So we have discussed about the organization of the genome organization in the prokaryotic cell, and we have discussed about how the different types of materials are required for genomic organization in the prokaryotes, what are the properties of the genome into the prokaryotic structures. We have taken an example of E. coli and how the compaction is happening inside the E. coli cell and so on. Now in today's lecture, we are going to discuss about the genomic organization into the eukaryotic cell and how the different types of chromosomes are being found. So in the genomic organization, when we talk about the genomic organization in the eukaryotic cell, we are having the genome which is present in two places within the eukaryotic cell. One is the nucleus and the second is the organelles. So within the organelle, you can have the two different types of organelle where the genomic content is going to be present. One is called as the mitochondria and the second is the chloroplast. So you can genome is actually been organized within the eukaryotes in the nucleus. So within the nucleus, you are going to have the different types of chromosomes, and within the mitochondria also you are going to have the mitochondrial chromosome and the chloroplast chromosomes, and summation of these DNA material is actually going to be called as the genome within the organisms. Now within the genome organization in the eukaryotes, so the eukaryotic genome is linear and confront the Western-Crick double-click helix structural model. It is embedded in the nucleosome complex DNA and protein structure that packed together to form the chromosomes. Eukaryotic genome have unique feature of exon-intron organization of the protein coding genes, representing the coding sequence and the intervening sequences that represent the functionality of the RNA inside the genome. A human haploid cell consists of the 23 different chromosomes, and one mitochondrial chromosomes contain more than 3.2 billion DNA base pairs. Remember that in the case of the prokaryotic structure, we were talking about the number of chromosome in the case of millions, but here the number of chromosomes are going to be in billions. That's why there is a more higher level of compaction is required, so that that particular DNA is going to be fit into the small tiny nucleus. Now as far as the chromosomes, so eukaryotic chromosomes are usually linear in structure. So this is the one of the eukaryotic chromosomes, and a typical chromosome is 10 of millions to hundreds of millions of bases pair in length. Eukaryotic chromosomes occurs in set. Many species are diploid, which means that the somatic cells contain two sets of the chromosomes, right? So you are going to have the chromosomes which are present in the sets, right? So double set is present inside the cell. During the duplications or during the division, one set is actually going to be shared with the daughter cells. Each chromosome contains a centromere that forms the recognition site for the kinetochore complex. So this is the centromere, right? This is the centromere. and this is the place which is actually going to be recognized by the kinetochore proteins. Then we have the telomeres, which contains the specialized sequences located at the end of the linear chromosome. So these are the telomeres, which are going to be 2 telomeres. So and this is the specialized sequences which are present on the tails or the corners of the chromosomes. Then we have the repetitive sequence which are found in near the centromeric regions. So these are the DNA or the genes, how the genes are actually being organized onto the chromosome. For example, this is a classical example how the MSC proteins are or MSC genes are being organized onto the chromosomes within, and you see that you have HLA typing and all that, and this is a particular type of chromosomes. What is the chemical composition of the chromosome or the chromatin? So you're going to have the DNA, you're going to have the RNA, and then you're also going to have the proteins. So you're going to have the DNA, which is 20 to 40%, so most important constituent of the chromatin. Then you're going to have the RNA, which is going to be 5 to 10%. It is associated with the chromatin as the transfer RNA, messenger RNA, or the ribosomal RNA. So it is actually be a part of the expression machinery. And then you're also going to have the proteins, so that is going to be 50 to 60%. So mostly the chromatin is actually made up of the DNA plus protein, and it is mostly the 40% is actually going to be DNA and 60% is actually going to be the protein. Within the protein, you're going to have the 2 different types of proteins. You're going to have the histone proteins or the non-histone proteins. So histone proteins, very basic proteins, constitute about 60% of the total protein. Almost it is present in the 1:1 ratio with the DNA, and you're going to have the 5 different types of histone proteins: H1, H2A, H2B, H3 and H4. Similarly, you're going to have the non-histone proteins. They are 20% of the total chromatin protein, and they are required for the nucleosomal assembly proteins such as NAP and the other histone chromatin remodeling complexes. And then you're going to have the structural proteins such as actin, tubulin and myosin, which and the contractile protein and all the enzymes. So the contribution of the non-histone protein is very little compared to the histone protein. Histone protein is required for packaging. So histone is a positively charged protein. So these are the basic proteins, which means they are actually going to be positively charged protein, and that's why they are actually going to have the instant attraction for the negatively charged DNA. So this is the DNA, right? You know that the DNA is negatively charged because of the phosphate backbone. So the histones are found in all the eukaryotic cells. They are commonly present histone in the eukaryotic cells are H1, H2A, H2B, H3 and H4. Then these all the 5 histones are being categorized into the 2 structural regions. One is called as the core histone, the other one is called as the linker histone. So the H2A, H2B, H3 and H4 are part of the core histone, whereas the H1 is actually being called as the linker histone. So core histone, the 2 copies of the core histone form the protein core around which the DNA is wrapped, and within this you are going to have the H2A, H2B, H3 and H4. Whereas the linker histone, it is not the part of the core protein, but it is associated with the linker DNA which links the 2 nucleosides. So within the histone you are going to have the core histone and the linker histone. The core histone you are going to have the 2 copies of the H2A, H2B, H3 and H4, which are actually going to form the core of the nucleosome, and then this core is actually going to wrap the DNA, right? So it's going to have the clear attraction because all these histones are going to be positively charged, so their surface is actually going to be positively charged, and that's how they are actually going to have the very high affinity for the DNA. Similarly, you're going to have the linker histone, which is going to be H1. So how these how histone proteins are, or what are the different properties of the histone proteins? So histones are closely associated with the negatively charged DNA. They have the high content of positively charged amino acids such as lysine and arginine. So you have the core histones and you have the linker histones. Core histone H2A, you're going to have the molecular weight of 14 kilodalton and it is actually going to be a lysine rich. Similarly, you can have the H2B, it's going to be approximately 14 kilodalton and it is the slightly lysine rich. Okay. Then we have the H3, H3 is going to be 15 kilodalton, it is going to be lysine rich. And you have the H4 which is going to be 11 kDa and it is going to be arginine rich. Then we have the linker histone which is H1 and it is going to be 20 kDa and it is also arginine rich. So H3 and H4 histones are first form the heterodimer, then come together to form a tetramer with the two other molecule of H3 and H4. H2 and H2B form the heterodimers. So this is the sequence in which the histones are actually going to be organized with each other, and that's how they are actually going to form the nucleosomal assembly. So the assembly of nucleosome involved the ordered association of histone with the protein DNA. So first the H3, the two molecule of H3 and the two molecule of H4 are actually going to come together, and that's how they are actually going to form a tetramer, which means you are going to have the like this, right? So you're going to have a tetramer which is going to be formed. Then this tetramer is actually going to, because this tetramer is going to have the positive charge, right? So it's going to have a positive charge on the top, right? And then it is actually going to bind the DNA. So this means this ball is actually going to have the DNA, okay? And then after binding the DNA, then the H2A and H2B is dimer, which means is actually going to join the H3H4 DNA complex, and that's how it is actually going to have the nucleosome. So this is actually one ball which is going to have the H3H4 DNA, and on top of this you're going to have the H2A, H2B, then H2A, H2B, like that. Okay, so DNA is actually going to be present inside this particular core. So this one copy of nucleosome is actually going to communicate with another nucleosome, right? So this is going to have another copy, and this is the region where the H1 is actually going to bind. So this is the linker region which is actually going to bind. The core histones have the amino acid extensions called as tail because it lacks the defined structural and do not participate in association of DNA with the histone octamer. The tails are the extensive site for a post-translational modification including the methylation, acetylation and the phosphorylations. The assembly of nucleosome involves the ordered association of the histone with the DNA. This is anyway we have discussed, right? H3H4 followed by the binding of DNA, followed by the binding of the H2A, H2B, and that's how the nucleosome is going to be formed. So the nucleosome is the starting building block for the higher order organizations. So nucleosome is actually the building block of the chromosome, right? So this is the DNA, right? Then it is actually going to form the nucleosome, then it is actually going to organized into the chromosomes, and then it is actually going to organize into the nucleus. So a human cell contains 3 into 10 to the power 9 base pair per haploid set of the chromosome. The average thickness of each base pair is 3.4 angstrom. This is the thickness of the DNA actually. So therefore, if the DNA molecule in a haploid set of chromosome were laid out end to end, the total length of the DNA molecule would be approximately 1 meter. For a diploid set, the length is actually the double, which is the 2 meter. As the diameter of a typical human nucleus is about 10 to 15 micrometer, it is obvious that the DNA must be compacted by many order of magnitude to fit into such a small space. The compactation in human nucleus is done by the nucleosome formation by the association of the DNA with the histone. Nucleosomes are packed into successively high ordered structures. So nucleosome model is scientific model which explains the organization of the DNA and the associated protein into the chromosome. It also further explains the exact mechanism of the folding of the DNA into the nucleus. This model was proposed by the Kornberg in 1974 and it is the most acceptable model of the chromatin organization. The model was further confirmed by the P audit in 1975. What are the features of the nucleosomal model? In eukaryotic DNA, in eukaryotes, the DNA is tightly bound to the histone protein, which leads to the formation of DNA protein particles called nucleosome. Histone play a very important role in packaging of such a long DNA molecule in the form of a nucleosome into the nucleus only a few micrometer in diameter. Therefore, the nucleosomes are called fundamental packaging unit particle of chromatin and it gives beads onto a string appearances, which means if you see very clearly you will see that the DNA is being packed like this, and this packaging is called as the beads on a string because this is the linker DNA and this is actually the core structure of the histone, and that's how these are actually going to be fold onto each other. So it is actually going to be fold like this, and that's how you are going to have beads, and then it's going to be fold like this, and that's how it is actually going to be keep condensing, and that's how you're going to have the higher organizations of the packaging. Each nucleosome is a disc-shaped particle with a diameter of 100 nanometer and 5.7 nanometer in height, containing 2 copies of each 4 nucleosome histones such as H2A, H2B, H3 and H4. This histone octamer forms a protein core around which the double stranded DNA is wrapped to 1.6 times, containing the 146 base pair. Each nucleosome bead is separated from the next by a linker DNA which is generally 54 base pair and contains a single H1 protein. This is what I was talking about, right? So you have a DNA and then it's actually going to be arranged. On average, the nucleosome repeat at an interval of 200 base pair. Folding of the DNA. So once the nucleosome is formed, there will be a folding of the DNA, and that's how you're going to have the higher compactation of the DNA. The assembly of DNA begins with a newly produced tetramer, which is H3H4, that is a particularly modified to form a subnucleosomal particle. The 2 HBA temers are then added. The results in the formation of a nucleosomal core particle with a 146 pair DNA bound to the histone octamer. The nucleosome is made up of this central component and the connecting DNA. In order to create the nucleofilament, the nucleosome core must be spaced regularly, which is accomplished during the maturation stage, which requires the ATP. The newly integrated histones are deacetylated in this stage. Next, the incorporation of linker histone is accomplished by folding of the nucleofilament into the 30 nanometer fiber, the structure of which remain to be elucidated. Two principal models exist. One is the solenoid model and the other is the zigzag model, actually to explain how the DNA is actually going to be folded after forming the nucleosome to form the higher order organizations. Last but not least, further subsequent folding process results in a higher level of structure and distinct domain within the nucleus. So these are the some of the organizations. So you are going to start with the DNA. DNA is actually going to be folded into the, so if you start with the 2 nanometer, then you are actually going to form the beads on a string form, and that's how actually you're going to have the nucleosomes. Then these nucleosomes are actually going to be organized and refolded. The DNA is actually going to be folded again, and that's how you're going to have the 30 nanometer chromatin fibers. Then these 30 nanometer chromatin fibers are actually going to be further organized, and they are actually going to form the 300 nanometer fibers, and in which these solenoids are actually going to be again folded onto each other, and that's how you're going to have these 300 nanometer fibers. Then this 300 nanometer fiber is actually going to be condensed, and that's how it is actually going to form the 1700 nanometer fiber, and you're going to have the condensed region of the chromosome, and from this you are going to have the further condensation, and as a result it is actually going to form the 1400 nanometer chromosome. So you are going to have the chromosome. So you started with DNA, you ended with the chromosome. So this is the first and this is the last binding stage. Okay, now in this this is the packaging of the genome into the eukaryotic system, and the genome has a very significant impact on the properties of that particular organisms, and the bigger the genome you are actually going to have, the more of the information what you are going to carry, and that's how you are actually going to have the higher flexibility of modulating that information, and that's how you are going to have the more properties to handle. So genome size is related to the complexity of that particular organisms, right? Because higher the information you have, the higher you are actually going to manipulate that information, and that's why you're going to have the complexity into the system. This means you're going to you can actually have the freedom to modify these proteins. You can actually be able to synthesize the proteins and so on. So if you see very clearly, what you are going to see is you are going to have the prokaryotic species and you are going to have the eukaryotic species. Within the prokaryotic species, you will see that the genome size is very small, and that's why the number of genes or number of proteins are actually going to be very small. This means they are actually going to produce the less number of proteins. And if you are producing the less number of proteins, you are actually going to have the lower order freedoms to manipulate those proteins, because you cannot have the multi-step process. You can have only the one or two step process, because if as many number of process you are going to produce, you can actually should have the proteins to regulate these steps. So that is basically a drawback, or I will say simplicity in the system, right? More and more you are actually going to have the genome size. For example, in the case of yeast, you are going to have the genome size which is 12 megabyte, right? And then you will see that the number of genes are going to be significantly very high compared to the mycoplasma. And more and more actually you see that these are the plant species, right? So they are very high, and the number of genes what you see here is very high. This means they are actually being able to have the potential of producing the large number of proteins, and that's how these large number of proteins can be utilized in such a way that you are actually going to have an event which is tightly regulated at each step, and that's how you can actually be able to have the more control over the process. So the C value or the COT values, or the quantity of DNA per haploid genome, such as that seen in the nucleus of a spermatozoon, is used to describe the genome size in the eukaryotes. Because the size is essentially consistent within the species, it is known as the C value or the characteristics. The mismatch between the C value and the presumed amount of genetic information contained within the genome is called C value paradox. Since we cannot assume that a species processed less DNA than the quantity required to specify its vital function, we have to explain why many species contain this amount of excess DNA. This is very simple actually. If you have excess amount of DNA, you can have the flexibility of producing more number of proteins, and that's why you can have the instead of having the 3-step process, you can have the 20-step process. Because if you increase the number of steps, you are actually going to have the flexibility. You have seen that the glycolysis is a 10-step process. Krebs cycle also has the multiple steps, and because they have the multiple steps, you can actually have the entry and exit of the metabolites at every stage, and that's how you are actually going to have the very complex biochemical reactions. Now the first question comes, if there is a requirement of the protein production, for example, if there is a requirement of protein production that is require that the DNA should be free for doing the transcription and translations. So now question comes, how you can be able to unpack the DNA, and how you can be able to have the DNA which is available for doing the other kinds of molecular biology activities such as replication, transcription and translations. So unpackaging of DNA. So the way we have discussed about the packaging of the DNA, the same way the histone have the crucial protein which are actually going to participate into the unpackaging of the DNA or the unbinding of the DNA. So histone are actually having the tail region, right? Remember that tail region is important for histone to be assembled with each other, and that's how they are actually going to bind the DNA. This tail region has the modification site for acetylations, for phosphorylations and the methylations. Now when you have the acetylation, you are actually going to produce the negative charge. When you have the phosphorylation, you are going to induce the negative charge, and when you have a methylation, that also is going to modulate the surface property of that particular protein. So as soon as you have the acetylation and phosphorylation, you are going to have the unbinding of the chromatin structures, and DNA becomes more accessible for the other kinds of downstream applications or other kinds of downstream activities such as replication, transcription and translations. So acetylations. So acetylation take place at the lysine residue of K4, K5 in the H4. It takes place through an enzyme which is called as histone acyl transferase or AT. The acetylated chromatins are more open. This means they are actually going to be active in terms of the replication and transcription. It is accessible for transcription factor and polymerases. Deacetylation takes place by the histone deacetylase or HDAC. The acetyl group donor is acetyl CoA. So you are going to have the closed chromatin, and when you have the activity of the histone acetylase, it is actually going to acetylate the chromatins, and that's how it is actually going to form the relaxed chromatin. Once the relaxation is over, when that process is over, then you are actually going to put the HDAC activity, which means the histone deacetylate, and then it is actually going to be closed chromatin, and that's how these are the things which are actually going to occur simultaneously, or to just to unbind the DNA, make it accessible so that you can be able to use that DNA, and then after that, once that process is over, then you can actually be able to close that DNA. Same is true for the phosphorylations. So you're going to have the kinase activity, which is actually going to convert the closed chromatin into the relaxed chromatin, and then you have the phosphatases which are actually going to remove the phosphorylations, and that's how it is actually going to reverse the events. Then third is methylation. So it occurs on the side chain of the lysine and arginine. The methylation does not alter the charge, but it actually changes the charge what is present onto that particular residues. So lysine can be monomethylated, di or trimethylated. Methylation done by the histone lysine methyl transferase and the histone lysine demethylase. So here also you're going to have the methyl transferase and the demethylase, and when you have the methyl transferase, the closed chromatin is going to be converted into the relaxed chromatin, and same is going to be reversed by the histone lysine demethylase. So these are about the normal chromosome. These are the information which is required for the normal chromosomes. But when people were discussing about, or when we started investigating the different types of chromosomes, they could found that some are the specialized chromosomes which is present in the some of the organisms. So let's discuss about these specialized chromosomes and how the DNA is packed into these specialized chromosomes, and what are the different properties of these specialized chromosomes. So the first is Polytene chromosomes. So what are Polytene chromosomes? Polytene chromosomes, which are gigantic chromosomes that grew from a smaller developing chromosomes, frequently appear into the salivary gland of Depterian flies such as Drosophila melanogaster. They are also known as salivary gland chromosomes because they were found in salivary glands. The Balbini found the Polytene chromosome in the salivary gland nuclei of the larva in the 1881. Due to the presence of several chromatin in them, they are known as the polytene chromosomes. Now, the question is how these polytene chromosomes are being found. So, the most polytene chromosomes are located in the interface nucleus of a few cells in the diphtherian fly larva. Each chromosome component is successfully duplicated as they grow from the chromosome of the diploid nucleus. After each DNA doubling, the later stage of the mitosis are eliminated, leading to the development of a polytene chromosome. As a result, the cell cycle is divided into the S phase and G phase. In Drosophila melanogaster, this polytenization cell cycle is developed during the mid-embryogenesis. DNA strands do not separate at the final stage of each S phase, rather they remain accompanied to one another, generating the polytene chromosomes. The process of endoduplications or multiple chromosome DNA replication without adequate chirokinases and cytokinases results into the polyteny of gigantic chromosomes. As a result, the giant chromosomes are produced, which are 70 to 100 times longer than the typical metaphors chromosomes. Morphological features of the polytene chromosomes. So, polytene chromosome is a very important thing actually, because this is a kind of an exception or kind of the structure what is being found in a specific organisms. So, there are numerous partially duplicated chromosomes that are almost intervened with each another, making up the polytene chromosomes. The heterochromatized centromere of all chromosomes fuse in a centromere. The polytene chromosomes are found in the form of 6 radiating arms from the chromocenter. You can have the X chromosome, you can have the 2 chromosomes, 2 chromosome left arm, right arm, 3rd chromosome right arm, left arm, then you can have the 4th chromosome, which is the shortest arm, then you can have the Y chromosomes and so on. So, an altered pattern of bright and dark is seen when these chromosomes are stained and examined under a microscope. Interband refer to a light pattern, which band refer to the dark pattern. So, this is a specific polytene chromosomes where you are going to have the right arm, you are going to have left arm, within the left arm you are going to have the left arm of chromosome 2, left arm of chromosome 3. Similarly, you can have the right chromosome of arm 3 and so within this place your centromere you are going to have the divergence, and that is how you are going to have the X chromosome and Y chromosomes and so on. And then you can have the some of the classical characteristics of these chromosomes, such as you are going to have the Balbini rings. The band undergoes morphological and biochemical changes related to their gene activity, and the activation of the genes of a band causes the compact chromatin strands to uncoil and expand outward, resulting in a chromosomal puff. The puff contains the DNA loops that are less condensed and the DNA of band elsewhere in the chromosome. Puffs are active gene of the transcription. So, these are the puffs, so chromosomal puff, and these are the active region of the gene expressions. What is the function of the polytene chromosomes? The nuclei of each cell enlarge in size, leading to a cell growth. The metabolic benefit of having a numerous copy of a gene allow for a higher expression of the gene expressions. The chromosome in Drosophila melanogaster undergoes numerous rounds of reduplications in order to generate the significant amount of glue prior to populations. The bar phenotype, which includes the kidney-shaped eyes, occurs from the tandem duplication of the severe polytene bands that are close to the centromere of the X chromosomes. Due to the fact that the polytene chromosomes are interface chromosomes and are thus transcribed, as a result, it offers a chance to investigate transcription by the direct observation, and transcriptional response to the certain stimuli can be observed. So, apart from the polytene chromosomes, you can also have the another kind of chromosome which is called as Lampbrush chromosomes. So, the Lampbrush chromosomes, what are Lampbrush chromosomes? Lampbrush chromosomes are transcriptionally active chromosomes which are mainly found in the germinal vesicle of large oocysts of many vertebrate and the invertebrate. The Lampbrush chromosomes derive their name from the lateral loop that extrude from the chromosome at a certain point. They are very much transcriptionally active as the emerging DNA from the certain point are rich in RNA polymerases. These chromosomes were first observed by the Fleming and Rickards in 1882 in oocyst of the amphibians. Where do these chromosomes occur? The Lampbrush chromosomes occur in the diplotene stage of the prophase 1 of the first meiotic division in the primary oocyst of all the animals, and the structure of the Lampbrush chromosomes. So, each RNA polymerase is attached to the nascent RNA and associated protein, generating the visible brush-like appearance. It can be visualized easily that Lampbrush chromosomes are held in a stretch out form during the diplotene stage of the prophase 1 of the first meiotic division. The axis of Lampbrush chromosome contain array of beads from which the loops are protruding outward, called chromosomes. They exist as meiotic bivalent to homologous chromosomes held together by the chiasmata. So, in this Lampbrush chromosomes, you are going to have the chromosomes, then you are going to have the RNA polymerase which is protruding towards so this is actually going to be region of single chromosomes and so on, and it is actually going to be transcriptionally very active because these are the region which are actually going to be available for transcription and the translation. So, they contain the symmetrical loops, one each other chromatin in a chromosome, their absence of lesion in the centromere region, each loop bears an axis which made up of single DNA molecule that is unfold during the RNA synthesis. What is the function of the Lampbrush chromosomes? Loops are useful in chromosomal mapping, then extremely helpful in visualization of gene expression and also the change associated with the transcription. It provides a great proof for the eukaryotic gene amplification which play a crucial role in the oocyte development, and it is helpful in the hybridization results. Now, at the end, we are going to discuss the comparison of the prokaryotic as well as the eukaryotic genome. Many of these properties we have already discussed, right? So, the comparison of the prokaryotic as well as the eukaryotic genome. So, prokaryotic genome is small in size, it is going to be large in the case of eukaryotic genome. Genome is going to be a DNA and a few protein in a simpler manner, whereas GNA in the case of eukaryotic cell, the genome is going to be present and many proteins are involved such as the histone proteins and so on. It contains a single set of chromosome, whereas in the eukaryotes you can have the multiple set of chromosomes. The amount of DNA is going to be small in the case of prokaryotic genome. It is going to be typically very large number of DNA which is present. Then the prokaryotic genome is polycistronic, whereas in the case of eukaryotic genome it is monocistronic. Then most of their DNA encodes for the protein, so it's actually there is no useless DNA, right? There is no DNA which is not going to be transcribed or which is not going to be translated to the protein, whereas most of the DNA does not code for the protein, right? It's a very small portion of the genome which is actually coding for the protein, rest all is non-coding regions. Then RNA processing not an option, right? So RNA processing allows for the several of these genomes because you have the non-coding regions. These non-coding region has to be separated from the coding region, and that's why the RNA processing is required. And messenger RNA has a short lifespan, whereas the messenger RNA is long life because the eukaryotic cell requires the continuous synthesis of a protein for several days, right? So these are the some of the properties of the genome. We have discussed about the prokaryotic genome and the organization of the prokaryotic genome, and we have also discussed about the eukaryotic genome and eukaryotic genome organizations. We have discussed about the organization of the proteins of the different proteins which are involved in the nucleosomal assembly and how the nucleosomal assembly is being formed and so on. So with this brief discussion about the genetic material, we are going to conclude our lecture here. Thank you.